Okay, welcome back everyone after lunch, and hope everyone had a nice break and maybe watched some of the last few talks for this last session of the SASAqS conference this year. And it's a bit of a general session with quite a varied talks from students on various topics related to aquatic ecology and aquatic sciences. So I think as there's so many different types of talks and different topics, I think we'll go through one by one, allow the presenter to give a brief overview, 2 or 3 minutes about the talk, and then we'll field any specific questions to that presenter. And then maybe at the end, if there's still a few questions for each presenter or general questions, we can field that as well. So first up this afternoon, we have Megan Reid, who's talking on the biological control of aquatic weeds in South Africa with a specific focus on Nymphia mexicana. Megan, over to you. Are you there?
I'm here. Can you hear me all right?
Yeah, we can hear you.
Great. Thank you very much for having me. So I'm Megan Reid. I'm from the Centre for Biological Control at Rhodes University. So I'm just presenting some work. It was based on a review done by a few of my colleagues and supervisors, just giving an overview of biocontrol of aquatic weeds. Many of you will have come across many of them during your work. And just before we continue, I'd just like to make sure we all know what biocontrol is, just in case anyone's not familiar with it. So generally, plants, when they're introduced into a new range, they become invasive because the natural enemies that are usually there to control the populations aren't there in the introduced range. So they can grow out of control, their populations become very problematic. And generally, as we've heard already from a few of the other talks this afternoon, they can be controlled manually or using chemical control. But unfortunately, this becomes very costly, it's very environmentally damaging, and it's also only effective in the short term. So with biocontrol, what we do is we go to the native range of the plants and we find insects or natural enemies that are well adapted to feeding and controlling those plants. We bring them in and test them to make sure they don't harm any of our native vegetation, and then we release them. So we kind of reestablish that balance to maintain control of those populations again. So historically in South Africa, we've had problems mainly with floating macrophytes and generally with what we nicknamed the Big Bad Five, which I'm sure many of you will be familiar with. So that's water hyacinth, red water fern, Kariba weed, water lettuce, and parrot's feather. And biocontrol was initiated in the 1970s, and we've had some really good success where 4 out of the 5 of these weeds are now being successfully controlled using biocontrol. But as I mentioned earlier, unfortunately, what we're seeing now is that as these floating macrophytes are being well managed, we're having a state transition. So we're finding that submerged and emergent vegetation is now becoming more problematic. So just to list a few, these are things like Iris pseudochorus or Egeria densa. I won't go into details here, but if you are interested, you can look at my talk. And we are investigating potential options for that biocontrol. But if you do have the time and if you have seen a few of these sites, of course, we'd really appreciate you letting us know, especially with these emergent plants, so that we can update our records, because we have sites popping up everywhere. And then finally, the plant that I'm focusing on is Nymphia mexicana, or Mexican water lily. So if you see a water lily, floating leaves rooted in the soil but with yellow flowers, it's not supposed to be there. And interestingly enough, during some surveys that I've done in South Africa, it seems that there's a weevil, a native weevil, that's formed a new association on this plant. So it shows a lot of potential that we could use this as an augmentative biocontrol agent, in addition to importing insects from overseas. So that's just a brief summary. Be very interested for any questions or discussions further. Thank you very much.
Thank you very much, Megan, for that introduction. Any questions for Megan? I'm not sure if there was any questions in the YouTube channel as well. Doesn't look like it. Maybe just from my side, Megan, how widespread is this Mexican water lily at present in South Africa?
Then so far, there are populations in Gauteng. It's quite prolific in the Western Cape. I've got a few sites in KZN and quite a few in the Eastern Cape as well. So it does seem to be quite widespread. And as I said, there are sites popping up all the time. So I'm sure we'll get even more records. And
the introduction was probably aquarium trade or something like that.
Yeah, a lot of these plants are very popular in the horticultural trade, but especially water lilies. And a big problem with this as well is that there are hybrids, so it makes things quite difficult in terms of finding agents that are well adapted to controlling specific genotypes. But yeah, certainly all these plants that are coming in, many of them are from the aquarium trade or horticulture.
Yeah, thank you. Any other questions for Megan? Okay, so if you do have other questions that pop up, we'll field them at the end of this session, and go check out Megan's video. Really interesting work on the biocontrol. Next up, we have Chad Keats talking about phylogenetic placement of a floodplain water snake. Chad, are you over to
Hey
you?
guys, yeah, can I go?
Yeah, you can go.
Fantastic. Do you must I put my camera on, or is it fine like this?
You can put it on if you want, or you can leave it off.
Okay. Okay, I am alive. Okay, so today I'll be talking to you guys a bit about my recent study. So I've just recently joined the wetland crew at Well. I'm a postdoctoral fellow working under Ryan Wasserman. And we recently went to Kruger National Park, well, to the northern reaches. And what we found, well, it's the first time I've ever seen it, and one of the rarest snakes in South Africa is called the Floodplain water snake, Lycodonomorphus obscurventris. And this animal had never been sequenced before, so it doesn't exist on GenBank, which is quite weird for a South African reptile because we've done quite well. And so for my study, I decided to sequence it and place it in the phylogeny of Lycodonomorphus. But to give some context, Lycodonomorphus are your water snakes. They are endemic group to Africa. Currently holds about 9 species, but they've had a very turbid taxonomical past. So for a long period of time, we had multiple subspecies, then morphotypes, then species. And for this species in particular, up until recently, it was considered a subspecies of a northern Malawian water snake. And based on morphology, they actually raised it to species level. And when I placed it in my phylogeny, I did a 4-gene tree. So I did 3 mitochondrial genes, 1 nuclear gene. And I put them together to make a maximum likelihood and Bayesian tree. We actually found that it was sister to the rest of Lycodonomorphus. So the common ground water snake was the most sister taxa within the group, and then after that came obscurventris, which is quite interesting because it was sister to the brown water snake, the dusky belly water snake, Witt's water snake, and to the common brown water snake. So it was very far removed from its original subspecies status. So it wasn't sister to Wittii, and which that shows that we shouldn't be basing all of our understanding on the taxonomy of reptiles and actually all taxa on just morphology alone. And when you start to incorporate a more unified species concept, when you delineate species and take into account multiple sources of information, you can really build better systematic groupings. In addition to that, what we found was that this species represented a 150-kilometer range extension north into Kruger, and it represents the most westerly range for this species and the most northerly for South Africa. And while we were there, we also managed to find other species that had never been found there before. So what this study shows both is that we don't know much even about our Southern African stuff. And the more we look, the more we're going to find. So as much as we think that we've kind of placed genetically, morphologically, everything in South Africa in the herpetofaunic groupings, there's still lots left to find, and there's still a lot to find, yeah. So that's my talk in a nutshell. Thank you so much.
Thanks, Chad. Any questions for his talk? Seems everyone is still full after lunch and a bit quiet. So, and I missed my connection dropped there, Chad. So just how widespread is this floodplain snake? Was it only occurring in the northern reaches of South Africa? Or?
It's quite widespread actually. So for the longest period of time, it was considered a synonym of Whittier, which goes all the way to Tanzania and Malawi.
Okay.
And it was characterized by two populations, obscure interest. So we used to think they were in KZN and Lower Kruger, and then you would find them in Zim, Mozambique, and up into Malawi. But with this new finding, it actually represents kind of a sample in the middle, which proves that the species is continuous all the way up into southern Malawi. So it's quite widespread, but because of its where it likes to live, which are these like floodplain regions, especially on the east coast, they're very, very hard to find, and they actually specialize in these floodplain systems, and you can't find them anywhere else. So we believe they eat frogs and largely fish. So because of this, honestly, their distribution, although massive, very few people encounter them, especially in South Africa.
And you said in Kruger you find them in what system there? I missed that, sorry.
Oh yeah, so it was the northern parts of Kruger, so that's the Makwaleka concession right at the top.
Yeah,
we found this one there. Yeah, previously the last specimen for Kruger was about 150 kilometers south of there in kind of central Kruger. So this mentions a huge range extension for Kruger. So they were very, very excited about it.
Oh, awesome. It's for everyone who hasn't been to that northern part, the Makuleki area, it's really amazing floodplain systems there. So really cool to get some cool results from them. Any other questions for Chad or maybe for Megan? Anyone? Doesn't seem like it. So thank you, Chad. We'll move on. If there's other questions, we'll field them as we go along.
Okay, fantastic. Thanks.
Next up is Shanae Kleinhans, telling us about environmental DNA methods, development for detecting Batrachochytridium dendrobatidis, or chytrid. Shanae.
Hello, Wynand. Can you hear me?
I can hear you. You can go ahead.
Sorry, my computer is just lagging, but okay, I'll continue. Good day, my name is Shanae Kleinhans. I'm a master's student at the Northwest University. Today I'll discuss my project on methods development for detecting Batrachochytrium dendrobatidis through environmental DNA. Batrachochytrium dendrobatidis is an aquatic fungal pathogen that has caused mass mortalities in amphibian populations globally. Conventional diagnostic assays are extremely invasive and involve the collection of toe clips, skin swabs, and mouth parts from tadpoles for analysis. Environmental DNA has previously been applied for pathogen detection as well as species detection in aquatic environments. Locally in South Africa, this research is still very young with no optimized eDNA protocol for BD detection yet. The methodology of this project consisted of two parts. The first part was the development and optimization of the protocol under laboratory conditions, and the second part was applying the protocol to field samples. For part one, the cultures were grown, quantified, diluted, and 3 culture types were prepared. 4 DNA extraction methods were then compared using these different culture types. 2 commercial kits and 2 crude extraction methods were used, and 2 of these methods were then selected for the next phase. A filtering protocol was developed and multiple parameters such as concentration, preservation time, and filter preservation mediums were tested. The results were quantified using nanodrop spectrophotometry and Tacman probe qPCR. Synthetic DNA was used to create a standard curve to which the zoospores were compared to determine the genomic equivalence, which was used to later quantify the results. This project has not been completed yet, and a couple of results still need to be obtained from filtering. After the protocol has been optimized, it will be applied in the Nkanda Grassland Nature Reserve. A fellow student at the Northwest University is currently working on the distribution of BD in the reserve, and these results will serve as the primary verification to the project. Amithia stellalandi, or the common river frog, occurs in abundance within the reserve as well, and special Amithia primers were developed to test on the eDNA samples. If this protocol fails to detect BD in the environment, the secondary verification will determine whether this is due to the protocol being insufficient or whether there's a lack of BD zoospores present in the environment. That's all.
Thank you, Shanae. Any other questions, or any questions for Shanae? Questions for Shanae, but it's really environmental DNA is really seems to be the buzzword, and it could work extremely well for disease detection. So really looking forward to all the results that come out from this.
Sorry, the connection broke a bit. I couldn't hear what you were saying.
I'm saying looking forward to the final results, and because I think that can be a really good detection system for diseases in aquatic ecosystems, especially something for chytrid that's so virulent, that damaging to our.
Yeah, I'm also very excited for it. If this protocol is successful, it can be applied to other aquatic pathogens in the future as well. And it's a less invasive alternative to most diagnostic assays.
No, definitely. There's a question for you, Shanae, from Megan Reed, asking, is this general pathogen affecting all frogs, or just in South Africa?
It affects all frogs, but some frog species are more prone or susceptible to it than others. For example, Xenopus laevis, or the plat anna, is, even though it can be infected, it's not affected by the pathogen at all. But other frog species experience mass mortalities from this disease.
You know, as far as I know, I mean, in other parts of the world, chytrid is a major problem for frogs and amphibians. So it's a great, definitely a disease of interest. Megan is also asking, just out of interest, do you know what the symptoms of infection are?
Yes, basically this pathogen occurs on the skin of the frogs, and it disrupts osmoregulation through the skin, which results in a buildup of toxins within the amphibians, and this is what ultimately causes their death. Yeah, there are many different symptoms. You can sometimes see it on the skin of the amphibians. Their writing reflex is sometimes altered. Yeah, it depends on the individuals as well.
Thank you. I see there's a question in the chat for Chad as well, and we'll just field it before we go on to the next presenter. From Matthew Burnett, says, thanks, Chad, a great talk. Just wondering around the habitat connectivity of these snakes and the distributions outside of South Africa, but around Mozambique and Zimbabwe. As these floodplains are not linked through a direct watercourse through Kruger, maybe linked through watercourses down the Mozambique floodplain. To what extent would species move over land to get to a floodplain habitat? As mentioned, the extension in Kruger National Park is a huge gap over some very dry regions. Chad, did you get that? It's a long comment / question. Not sure. I think we've lost Chad there, for instance. Anyway, we'll come back to that question, Matthew. A really good question, I think, about habitat connectivity for these snakes. So we'll move on then to the next presenter, is Lizanda Nacker, on the current and future predicted status of schistosomiasis in South Africa and the role of climate change. Lizanda.
Hi, Wynand.
There, you can go ahead. We can hear you.
Great. So this between Northwest University and University of Limpopo, and it's funded by the WRC. So the overarching aim of the study is to update the records of schistosomiasis snail vectors in South Africa, because the last really comprehensive research was in the 1960s and 70s. So what we first did was digitize the Bilharzia Atlas of South Africa, and that is now available online for the general public. It's still a bit of a rough draft, but we will improve it as the project continues. What we are doing from here is we are using the historic distribution sites for the vectors and determining if and how their distribution has potentially changed over the past 2 decades. This is accomplished using changes in climate, particularly temperature and rainfall, and human population growth, because that's those are some of the factors that are the most predominant effects or factors that affect schistosomiasis distribution. We've also started collecting snail samples from the historic distribution sites to see not only if the snails and parasites are still where they used to be, but also if any other snail and parasite species that were not previously reported there, especially the invasive ones, maybe are present at the sites now. This is pretty important because some of the invasive snails like Cerebia granifera were not even present in South Africa yet when the snails were originally collected. Invasive snails can have a huge impact on the native distribution of species, so that could also then affect schistosomiasis distribution. We will also be collecting snails from potential new distribution sites based on the models to see if it is able to predict the distribution of the snails, and then if the snails are present, whether or not they actually have any of the parasites as well. So this work forms part of the MSc student Herman's project, which he presented in a poster here at the conference. So please go check that out if you have the chance. Just plugging quickly. Once the kinks in the model have been worked out, we'll also try to model how the snail and vector distributions change in the future. An additional section of the project is looking into the knowledge, attitudes, and perceptions of people living in areas with high schistosomiasis prevalence. So that is where our colleagues from UL come into play. Farina Lindicki and her MSc student Nape have, along with a bunch of honor students, done over 300 surveys with the local community, asking them questions about their knowledge surrounding bilharzia, if they know what it is, the cause, the symptoms, etc. We only have some of the preliminary results so far, but it's nice to see that most people do indeed know what bilharzia is and what the symptoms are, and that medical treatment should be sought. They also did know what climate change was, but most people don't really know exactly what causes bilharzia. They seem to think it's some kind of bacteria or virus. So just at the end of the project, we'll also have a workshop with this community to show them some of the results that we found, show them what the snails look like, give them posters to put up in local clinics and community centers with information about bilharzia, and just try to help educate them in areas that we saw a lack of knowledge from the surveys. Yeah, that's me.
Thank you, Lazan. Questions for Lazan?
Yes, I see there's one from Matthew in the comments. I have to, up to this point, no one that has gone into the field have gotten bilharzia, so we're trying to avoid that at all costs because we wear waders and try not to touch the water too much.
I think one of the symptoms of bilharzia is being sleepy and tired after lunch. So I think many of the people here in the session.
No, that's sleeping sickness.
No, also asking in the chat where we can access the atlas.
So I have the link for the website on my presentation. Otherwise, I can also I'll plug it into the chat box as well. I'll just have to go onto my laptop.
Okay, thanks, Lazan. That would be great to just. There's a hand from Chris Curtis.
Hi, Lizania. Another question on bilharzia. I just, sorry, I haven't watched your video yet, but I am definitely going to. I'm a little behind. But my daughter caught bilharzia at Bronkhorst Spray Dam a couple of years ago, and I've been told that studies have been done there on the distribution of bilharzia in Gauteng, and it wasn't found. I just wondered if you knew anything about that, because the rowing community who use these dams in Gauteng, I have a lot of anecdotal evidence that many of the rowers are being diagnosed with the bilharzia, and it seems to be undetected by the scientists. Do you have any comment on that?
I also actually received an email recently saying that they've heard about people getting it in bronchosprate. So it is something that needs to be looked at. I'm not sure exactly which methods the people who assess the system used, but I know if you just do the saccharial shedding in the snails, then your chances of actually detecting the parasites is quite low. So other methods might have to be used. We're also actually looking into that, like using eDNA and also taking actual tissue from the snails and then doing DNA analysis to pick up if there are any parasites in the snail tissue that were not detected with the normal screening methods. So it is definitely something that needs to be looked at. I mean, if people are getting it there, it has to be there somewhere. I don't know if they actually found the snails, but if they're getting bilharzia there, then the snail should be there, even if it's low abundance.
Thanks. Can I just respond? I was told by the catchment forum, Cara Stokes, who runs the catchment forum at the Bronchies, she said that CSIR had done a study, and I think they'd failed to find the snails, which is a problem. So I think there's clearly still a lot of work to be done.
No, definitely. I mean, the snails can sometimes be in very, very low abundance as well. So, no, I don't really know what else could be added.
But yeah, I think Lizanne is definitely something maybe that we can look into if we drive past or at a closer site. There's a hand from. Sorry, you wanted to say something, Lizanne?
Oh no, I'm done. Sorry.
A hand from Matthew Burnett.
Yeah, thanks, Fernand. Thanks, Lizanne. No, it's very important work what you're doing, and like with the rivers that we work in here, it's pretty much given that at some point in your career working here you would get bilharzia, and so we just almost every year if we feeling the symptoms, we take the medication needed to get it out of the system. And I think just adding to Curtis's thing, maybe, yeah, I think his paddler should come paddle the doozy for a while, and then they'll realize how it kind of comes with the territory of being a paddler here. So as much as these are important studies in terms of disease management, it's very much a way of life where we are. So
yeah, that is true as well. You have to, if you know about bilharzia and you know there are risks involved, then it comes, as you said, it comes with the territory. But if there aren't any specific indications that bilharzia is present in bronchosprate, maybe a sign should be put up at least just to warn people, you know.
Yeah, that doesn't stop paddlers in the doozy.
No, I can imagine it doesn't stop most people.
There's also a hand up from Ryan Wasserman. Ryan, maybe you mute.
Hi. Thanks very much, Lizanne. I just wanted to find out, and I haven't listened to your presentation yet, but have you guys looked into using eDNA techniques for sampling, especially in those systems where you're not finding snails themselves in high abundance?
Yes, actually, we are looking into using eDNA. At the moment, because we've been struggling to detect them in the snails themselves, so we've been taking DNA, tissue from the snails to try and detect the actual parasites in the snail tissue. But yes, we are actually looking into eDNA. Vainant and I actually recently did like a test run with some eDNA kits for in other systems, and was just waiting for some of the results. I don't know, Vainant, if you have any specific additions to that section.
Okay, thanks. Is that a new hand, Ryan, or still the same one?
Sorry, that was the same hand.
Yeah. I see Mike Silberbauer also added the link to the Atlas Design project. So Patsy, the link is there for you to have a look at the preliminary atlas and that we're working on. Any other questions for Lizanne? Okay, so we'll move on to in the program. You'll see there's 3 talks on invasive crayfish in Africa and the Zambezi Basin. 2 from Takudzwa Madzivanzera and 1 from Elise Streeter. Takudzwa could unfortunately not make the session today, but his videos are up on YouTube. So if you have any questions for him, please leave it in the comment section and he can answer at his leisure. So I'd like to ask Marlies Streeter to tell us a bit about the co-introduction of temnocephalin ectosymbionts on the crayfish from Zambia. Marlies, are you there?
Hi, Wynand, thank you. I'm here.
Yeah, you can go ahead, we can hear you.
Okay, so my presentation is just a brief summary on findings of data that we collected when an opportunity presented itself. And I have to thank Takudzwa and Josie for bringing our attention to the infection during their sampling in the Barotse floodplain. So thank you guys, if you're there. If you have watched Taku's presentation, you would know the extent of the distribution of the invasive red claw crayfish in Southern Africa, but if not, it has invaded Southern Africa's fresh water and it has socio-economic and ecological impacts, and this includes the co-introduction of parasites and diseases. So currently we have an idea of where the crayfish occur and where they are spreading, but we have very little data, or actually none at the moment, 1 report of successful co-introductions and their parasites or ectosymbionts. But we don't know if they're posing a threat to native crab species. So currently the main focus is on this group of platelmints known as the temnocephalins, and we know of 3 species in Southern Africa that were co-introduced with the red claw crayfish. That's in the Komati and Pongola systems. That was close to the point of introductions initially. So a 4th species from a different crayfish species, the hairy marron, has been proven to switch host to native freshwater crabs in laboratory conditions. And then this temnocephalin also acted as a vector for pathogenic bacteria. So that caused the death of the crabs. So in short, during our study, we identified the species that were present in the Barotse floodplain using morphological and molecular techniques. And we also identified that there is, or came to the conclusion that there is no research that track or investigate these co-introductions or spread of these temnocephalins, as well as their behavior with their host, or the association they have with the freshwater crab hosts in Southern Africa. Because there is a possibility that some of these temnocephalins feed on the eggs of their hosts, that can have detrimental consequences if our native decapods, or the crabs or the decapod group, are susceptible to these temnocephalins. And lastly, the presentation then emphasizes that all of us that works closely with freshwater species, we must report observations of infections on both invasive species as well as on native species. And we should report, or must, observations, oh, I already said that, sorry. And we also must attempt to collect the specimens where possible. It is quite easy to collect these temnocephalins. Some of them are big if you watch the presentation. Some of them are quite big. So if you just brush them off and pop them in ethanol and send them off either to someone that you know that work on temnocephalins or that might know what it is, or me, then we can actually just keep record of where they are and if they have been spreading to native crab species or other decapods, and if they are affecting these populations. Yeah, thank you.
Thank you, Marlies. Any questions for Marlies from the audience? Seems there's no questions for Marlies. Marlies, maybe at present, do we know of any of these temnocephalans that has been found on our indigenous crab species? Or not?
Vainan, so only in that one experimental or that laboratory experiment proved that it can transfer to native crab species, but no observations has been reported from native populations. So I don't know if that is because there's a lack of actual investigation, or if people see it that they're not curious enough, or yeah, I don't know.
Okay, no, it's probably got no observations yet, guys.
Yeah.
Other questions for Marlies, or if not, there was a question for Chad on the habitat connectivity that Matthew Burnett posed. Chad, are you back now?
Yeah, sorry, I'm back now. Can you hear me?
Yeah, we can hear you.
Fantastic. So yeah, that's a great question actually, and it's something that I want to look into in more depth. It's a phenomenal phylogeographic question to determine the connectivity of these animals, because anecdotal evidence suggests that they actually do desiccate outside of water. So you take into consideration the floodplain water snake and especially dusky belly water snake. If you remove them from water for a long enough period of time, they will start to dry out. They'll actually start to notice that their rostral scales will actually dry out and they actually start to desiccate. So the ability to navigate terrestrial habitats to get to new sources of water, it's hard to say, but I can't imagine it'll be very far. But it's a question I would love to answer in the coming years. I've definitely got a bee in my bonnet for Lycodonamorphus now. Getting samples of bicolor and seeing the connectivity between the different populations it's a great question, and I agree with you in terms of Mozambique and Zim. It's just in Kruger, what is going on? So great question, and I would love to answer in the next few years. Thanks so much.
Okay. Any other questions for any of the speakers in this session? If you have other questions, if you go and watch all these videos on YouTube, on the YouTube channel, please do so, and pop any questions into the chat box in the YouTube comment section, and we'll ensure that, or hopefully the authors will be able to respond to you. So if there's no other questions, let's take a 5-minute break. Let's start at 2:20 with just the last few closing remarks and the awards. So I'll just give everyone a chance to stretch your legs for about 5 minutes, and then we'll start at 20 past. Thank you.
Yeah.
Okay, I think let's start. Seems like everyone that's going to join is joining now. So we've now come to the end of the SASAqS 2021 program, and from my side, I think it's been a really interesting and engaging conference with a lot of discussions, a lot of interesting discussions on the various projects and sessions that we've had. And I, for one, have really enjoyed all the discussions and the different presentations. And I would like to thank everyone for your participation and your involvement. And for everyone who hasn't had a chance yet to go and look at all the amazing videos and presentations on our YouTube channel, please do so. They will remain up for at least till the 15th of November. If there's people that would like to keep it up longer, please drop me an email and we will arrange that as well. So please, I do encourage you to go and look at the YouTube channel. And then I would like again to thank all of the sponsors for this year. Nkaba Biotech Separations, Symbiolab. Thank you very much for your support of our conference, and we really, really appreciate your contribution. And then again, I would like to thank my team behind the scenes, Kerry, Hannes, Audrey, Chantal, and Marilise. Thank you very much. Without your hard work and help and support, this would definitely have not been possible. So again, from my side, thank you very much. And so first up, I see Richard has asked Leon if he would like to tell the members about next year's conference. But before we get to Leon, who's going to share us just a little information about next year's conference, I would like to ask Lizanne de Nacker to announce the various winners of the student prizes. And as she was the chair of the judging committee, helping with, and there was a few early career and postdocs helping her out with this. I'm pretty sure was a very difficult task to go through all the presentations and decide who was the best students' presentations for this year. So, Lizanne, can I hand over to you?
Hi, Bernard. Yes, you can hand over to me now. I'm going to share my screen, and then you need to tell me if you can see the right one. This one. And then, woo.
Yes, we can see that.
Okay, which are you seeing my view or are you seeing the full presentation?
We're seeing the presenter view.
How's that?
Yeah, now it's full on the screen, so you can go ahead.
Okay, great. So first of all, I would like to thank my fellow judges who helped with figuring out which student presentations and posters were the best. It really was a very difficult decision to make. So thank you very much to Simone and Matthew and Jeffrey for assisting me in judging. And then I would also I just want to give a very big thanks to all the students for their presentations and posters. The quality really was very high, and myself and my fellow judges struggled to find the top presentations. Now, before I move on to the prize giving section, I just thought that it would be nice to just give some general comments and suggestions for all the presenters for the future, and make it even harder for the next judges to decide which presentations are the best. So, yeah. All right. So some general tips are that in the future, if you do have a pre-recorded presentation, at some point, please make sure to listen to the recordings once you have finished them. Sometimes people spoke either too close to the microphone and the sound was very distorted, and other times people spoke so softly that it was really difficult to hear what they were saying. A general issue that also occurs in all forms of presentation is a speaker that speaks too quickly. This happens because of nerves, I understand. I struggle with it myself as well. So it is something that we all need to work on, because sometimes people spoke so quickly, it was really difficult to follow what they were saying. So rather just take a moment, slow down, take a breath, make sure everything that you're saying comes across clearly. Then, when it comes to animations, really less is more. Keep them to a minimum. You really should only keep animations to a minimum and use them when they are necessary and add value to your talk. Bullet points that fly in individually should be avoided at all costs, because there's really no point in having so many animations if you're going to be spending the first 10 seconds of your presentation having each one fly in individually. It really takes up a lot of time, and it doesn't add a lot of value. Next, when it comes to color, there's really nothing wrong with using color in your presentation. It can add a lot of value. It can highlight some nice particular sections or words, and it just makes it nice and pleasing to look at. But you have to make sure that it's legible. Otherwise, it takes away from your presentation. If you're using a black or very dark background, a white text or a color that is very close to white will be your best bet, because it really contrasts the best. Light or bright colors can also work, but you have to be careful using too much color, because it can make your presentation seem like a disco rather than professional. So the general rule is to pick 1 or 2 colors that work really well with your background and stick with that throughout, and make sure that your color doesn't blend in with the background. If you have a white or very light background, black text or a very dark text really is the best way to go. Here, very light or bright colors don't really work. They blend in with the background, so you have to try a more subdued, darker color. Or you can use a colored text box with white or black text. But again, you should avoid overuse. And again, also make sure that your color doesn't blend in with the background. You can also make your text much easier to see and read, and add emphasis by putting it in bold where necessary. In terms of pictures, clip art and images really can add a lot of value to your presentation, but there are certain etiquettes to follow. Don't stretch your pictures out to the point where it distorts what it was supposed to be. You can add a border or two or something nice if you really want, but if you use a bunch of funny different styles, it really takes away from the image that you wanted to use. And also, if you have a lot of pictures, be careful to not make your presentation too overcrowded and have figures that overlap one another, and it also takes away from the value. So rather take that extra minute or two.
Okay, channel next is about 1.5 to 2,000. Ntoki chelete ntona yo tla go wena o tla kwa botse. Maximum even 2.
Okay.
Yeah, I think you can go ahead, Lizanne.
Hello?
Je kan aangaan, Lizanne.
Kan jy my hoor, Wynand?
Ek kan jou nou hoor, ja.
Well, okay. Anyway, sorry. All right. Finally, my final comment is about the amount of text and use of space on slides. When you're facing a wall of text like this, it really can be very intimidating, and the text can be very small and difficult to read and focus on. It also makes the slide seem very crowded, and even though there's a bunch of open space, there's a lot of space being wasted. So rather just break your sentences into smaller, manageable sections, maybe over a few slides if there's not a specific limit to your number of slides. And in this way, you can make your text larger and easier to read, and even though there's still space around that is not used, the presentation seems less crowded, and maybe a picture or two can be added somewhere if necessary. So with all that now in mind, I would just like to move on now to the actual reason why everyone's here for the presentations, the best presentations and posters. First, I would just like to give a few special mentions to some of the students who really came so close, because it was such a difficult decision to make. So congratulations, Casey Broom, Tolwana Mtokwane, Hima Rama, Liam Connell, and Chad Keats for really making it one of the most difficult decisions. You were in the top choices there. Then I would like to, for the third place presentation, congratulate Heinrich Doms for his presentation on fantastic plastics and where to find them. If you haven't watched his presentation yet, please do yourself a favor and go do that. It's very interesting, but also entertaining. In second place, I would like to congratulate Tembela Vimbi for her presentation on the role of disturbances in shaping microbial communities in the Nahoon estuary. It was also a very informative, very nice presentation, so congratulations to you. And then finally, the first place presentation is for SASAqS for her presentation on morphologic deformities in Oreochromis mozambicus during early life in response to chronic exposures to this efavirenz. It was also, I did watch the presentation. I'm sorry I can't properly enunciate the word, but it was a very nice presentation to watch as well. So congratulations to you. And then for the best student poster, there is just one prize, but I also just want to give special mention to 3 students, Martin Herman Leroux and Alicia Riley and Mbahle Le Ngobeni. You guys really made it to it was between you 3 and the person that won, so congratulations for almost winning. But the best poster presentation goes to Greg Janssen van Rensburg for his presentation poster on median lethal concentrations of 2 pesticides in the freshwater shrimp, Caridina africana. And with that, just thank you very much, and over to you, Wynand.
So I feel we will be able to
Thank you very much, Lizanne. Having watched Many of these, most of the presentations already, I can assure you it was not an easy task to announce these winners. So thank you, Lizanne, and congratulations to all the winners. Job very well done, and we'll communicate with you regarding your prizes in the next few weeks. So keep a lookout for an email from me. So I'm sure everyone, or I hope everyone enjoyed this year's conference, but I'm sure I speak on behalf of many of you that we are really looking forward to an actual in-person SASAqS conference that will be hosted next year. And to tell us a little bit about their plans for next year, I would like to introduce Leon Barkhuizen to announce the SASAqS 2022. Leon.
Hi, Wynand. I'm just trying to share my presentation. Can you, okay. Can you see that?
Not yet.
And now?
No, not yet.
It's on my screen. Is it not on your screen yet?
No, unfortunately not on mine.
I wonder, let me try again. Is it there now?
No, not not yet. Sorry.
Do you still have it, the email I sent you? Can you maybe share from your side then?
Yes, I'm gonna just give me 2 minutes and then I'll share it.
Okay.
Just 2 minutes. Sorry for the delay, everyone. Just gonna share the screen in 2 minutes. Okay, sorry about that, everyone. Like half an hour ago, we had a massive Wi-Fi failure and we had to swap computers. So yeah, just scrambling a little, but I have Leon's presentation. So Leon, are you ready?
I'm ready when you're ready.
Okay, I'm sharing it now. Can everyone see?
Right, thank you very much. I think first of all, Wynand, I really want to thank you and your team, and I think all of you really deserve the bells this evening. Thank you very much. I think to host such a conference is really a challenge, but you've done really great, and thanks for you and your team's great effort and all your hard work. I'm presenting on behalf of Lisa van As, who unfortunately must attend a faculty meeting this afternoon. So as part of the organizing committee, we would like to invite all of you to the SASAqS 22 conference, which will be held at Zambezi Private Game Reserve in the Free State, near the town of Winnie Mandela. For those of you that do not know or heard about the town, it's a previous Brandfort. Next slide, please. Right, the local organizing committee is the University of the Free State, Department of Zoology and Entomology, Liesel van As, Candice Jansen van Rensburg, and myself. And then also from the Center for Environmental Management, we are having Paul Gobelhortzer and Marinda Avenant. If anybody at this stage, I know we are plus minus 7 to 8 months away from the next conference, but in the meantime, if you have any queries, you are welcome to send an email to Liesel van As at vanasll@ufs.ac.za or to Marinda Avenant at avenantme@ufs.ac.za. Next slide. Right. Zambezi Private Game Reserve is the venues for conferences, weddings, and private celebrations, and it caters for groups from 15 up to 300 guests. For those of you that you know must start your planning, the average distance from the Bram Fischer Airport in Bloemfontein is 80 kilometers from Bloemfontein Airport, Johannesburg 360 kilometers, Polokwane 670, Geberga 710, Cape Town 1060. If you are from Timbuktu, it takes you 9,000 kilometers traveling to get to our conference. So that gives you an idea how far and how long you'll have to travel to come to our conference. Next slide. Right, there are various venues. There's a conference hall. I can seat up to 80 guests, but I think we are really looking forward to really meet all of you face-to-face and have face-to-face discussions and all the nice gatherings in the evening. So that first conference hall is maybe not what we're looking at. There's a nice bush boma, and you'll see later we're going to have one of our functions there. And there's now a nice dancing area between the tables and the fire. So yeah, we'll have lots of fun with these excellent venues at this conference facility. Next slide. At this stage, we have decided on the hall. You can see the picture there. That's the main conferencing hall. It can seat up to 300 guests seated at the tables, or 400 guests in a cinema style. Next slide. Next one. Are we stuck now? Right, for those of you that's going to do talks next year, we are sending you a forewarning because there's a very unique setup in this main conference hall. There's a podium in the middle of a conference room with a TV screen in front of a speaker. Seating are to the left and to the right of the podium with a large screen in each section. And the pictures below were taken from the podium. You can see the left view and the right view of the hall. After the plenary sessions, the hall can be divided into two with steel doors in between for breakaway sessions. There's also another option for breakaway sessions in a sitting room, but it's just above the hall. So yeah, prepare yourself for a unique conference setup and a nice experience in this Sable Hall. Next slide, please. Right, there are a wide variety of accommodation options available, from mountain chalets. If anyone wants to get married in June and celebrate with us, there's beautiful honeymoon suites, a leopard rest lodge, family chalets, guest houses for self-catering, cottages, single rooms, dormitories. There's also chalets for paraplegics, and there's a caravan park, but we will not recommend any caravanning or camping because it's winter time in the Free State, so yeah, I won't recommend that. Next slide, please. There's a wide variety of activities to enjoy at Amanzi. There are game drives, archery, horse riding, paintball, walking trails, and mountain biking. And there's a possibility of a surprise activity or activities that we are currently planning as an organizing committee. Next slide. Right, we have set the preliminary dates. It's 26 to 30 June 2022. And the theme of our conference will be valuing our aquatic ecosystems, source to sea. With all the previous face-to-face conferences, we start on a Sunday, the 26th, with arrival, registration, and meet and greet in the evening. Monday evening or Monday, we'll have a conference in daytime, and then in the evening, we will have a bride, a real Free State bride in the Boma, dancing around the tables around the fire. Tuesday, conference daytime, in the evening, informal dinner for all. Wednesday evening, Wednesday conference, and as usual, the AGM. And we're going to all you in a gala event in an awesome venue. Thursday, departure and check at 10 o'clock. And then there's an option for various workshops, which will be confirmed. The registration fee to be confirmed early 2022, but it will be an all-inclusive price or package. It will include accommodation, all meals, conference goodies, and registration fee. So we would like to welcome all of you. We are really looking forward to have a face-to-face conference. We've got an excellent, beautiful venue, and we really hope to see all of you at Amanzi Conference Center in June next year. Thank you very much, Wynand.
I'd absolutely recommend.
Thank you very much, Leon. Really looking forward to some Free State hospitality next year at the conference. So maybe if there's anyone who'd like to make a final few questions or comments, if there isn't, we can close SASAqS 2021. And I see there's one hand from Richard.
Thanks, Wynand. Just from the EXCO side, I'd like to thank you and your organising committee for organising this conference. I don't think it's been an easy task, and there have been a number of technical issues, and I think you guys have coped with it extraordinarily well. So well done for that. And yeah, I think the presenters have all presented really well. I think the video format has worked nicely with the option of going back to see presentations if you weren't able to see them in the beginning. And all in all, I've had an enjoyable conference, and I think you know, the discussions have been good. So from an EXCO's point of view, I think you guys have done very well, and I think you deserve a large round of applause. Thanks, guys.
Thank you very much, Richard. Yes, so thank you very much for everyone's participation. It's been great. Go and watch the YouTube videos, go and like them for the next 2 weeks. We will announce the winner of the most liked video in via email, so, and maybe a video message. So enjoy the rest of your day. Thank you for participating, and see you in the Free State next year. Bye, everyone.